Good evening, viewer. Welcome to our Glory Globe TV. Here's the most amazing news and breaking news for our viewers. Researchers have discovered an antibody able to neutralize all known variants of SARS-CoV-2. Yeah, the virus that causes COVID-19 as well as distantly related SARS-like coronavirus that infect other animals too. As part of a new study on hybrid immunity to the virus, the large multi-institution research team led by the University of Texas at Austin discovered and isolated a broadly neutralizing plasma antibody called SC27 from a single patient using technology developed over several years of research into antibody response. The team led by UT engineers and scientists obtained the exact molecular sequence of the antibody, opening the possibility of manufacturing it on a larger scale for future treatments. Yeah, the discovery of SC27 and other antibodies like it in the future will help us better protect the population against current and future COVID variants, said Jason Lavender, a research assistant professor in the Cockrell School of Engineering, Maketa Department of Chemical Engineering, and one of the leaders of the new research which was recently published in Cell Reports Medicine. During the more than 4 years since the discovery of COVID-19, the virus that causes it had rapidly evolved. Each new variant has displayed different characteristics, many of which made them from resistance to vaccines and other treatments. Protective antibodies bind to a part of the virus called the spike protein that acts as an anchor point for the virus to attach to and infect the cells in the body. By blocking the spike protein, the antibodies prevent these interactions and therefore also prevent infection. Yeah, SC27 recognized the different characteristics of the spike protein in the many COVID variants. Fellow UT researchers who were the first to decode the structure of the original spike protein and paved the way for vaccines and other treatments verified SC27's capabilities. The technology used to isolate the antibody, termed IG sequence, gives researchers a closer look at the antibody response to infection and vaccination using a combination of single cell DNA sequencing and proteomics. One goal of this research and vaccinology in general is to work towards a universal vaccine that can generate antibodies and create an immune response with broad protections to a rapidly mutating virus, said Will Foss, a recent PhD graduate in cell and molecular biology in UT's College of Natural Science, who co-led the study. In addition to the discovery of this antibody, the research found that hybrid immunity, a combination of both infection and vaccination, offers increased antibody-based protections against future exposure compared with infection or vaccination alone. BNS exclusive report, Glory Globe TV, Delhi, Bharat.